DNA.
DNA. But what is DNA actually? You might be thinking,
Oh, come on, Melek, be so for real. This is DNA.
and while you're not completely wrong, I'd like to tell you that this is not the full image. This is what DNA actually looks like. DNA, or deoxyribonucleic acid, is the complex molecule that contains the complete genetic instructions for the development, functioning, growth, and reproduction of all known organisms and many viruses. It is essentially the master blueprint of life. Do you see those shapes on the right side of the screen? Those are called nucleotides. So nucleotides are actually what make up DNA. And now you're of course asking, what is a nucleotide? This is a nucleotide, and it is the fundamental monomer or building block of nucleic acids like DNA and RNA. It is a small, complex organic molecule that serves as the basic repeating unit in a long chain structure of genetic material. Every nucleotide has three chemically linked components. Number one is the phosphate group, and number two is the pentose sugar. What interests us most today is the third component. You might be asking,
Why?
and to that, let me tell you that it is the component that provides the genetic code, and it is the nitrogenous base. As you can see, this nitrogenous base is called adenine, and there are four main bases: adenine, thymine, guanine, and cytosine. And in short, we call them G, A, T, and C. The DNA structure relies on a strict pairing rule. Adenine A always pairs with thymine with two hydrogen bonds, and guanine G always pairs with cytosine C with three hydrogen bonds. And a single DNA strand is connected together by phosphodiester bonds. This is what gives DNA its helix shape. Since DNA is not today's topic, we're going to use a more simplified structure, which is this. And now I'll provide you with an image, and you'll try to guess today's topic. Any guesses? Drumroll, please. Today's topic is CRISPR, gene editing technology.
This baby just became the first person in the world to fix their DNA through personalized gene editing. Let me explain. Scientists used a protein directed by molecules that act like GPS coordinates to search through a cell's DNA until it finds a match and then makes a cut so they can stick in new DNA. This technology is like a text editor for genes. And this baby had one tiny error in its DNA that was causing a life-threatening disorder. So they fixed that one error out of 3 billion DNA pairs. What that means is that we can now identify the genetic markers for sickle cell or Huntington's or certain cancers, and then disable or replace them. And the sickle cell example is happening with live patients right now. If you need more optimistic science and tech stories, subscribe.